Metabolism refers to all the chemical reactions that occur in living cells. The cells of your body use molecules of adenosine triphosphate, ATP, as their primary source of energy. How does the cell produce a continual supply of ATP? The answer lies in the process called cellular respiration, which occurs partly in the cytoplasm and partly in mitochondria. During cellular respiration, a glucose molecule is completely oxidized. In the process, it releases enough energy to produce up to 36 molecules of ATP. The first phase of respiration, glycolysis, occurs in the cytoplasm. During glycolysis, the 6-carbon glucose is broken down into two 3-carbon pyruvic acid molecules. Glycolysis also generates two ATP molecules and two molecules of an electron carrier called NADH. Pyruvic acid can be metabolized in two possible pathways, depending upon whether or not oxygen is present. In the absence of oxygen, pyruvic acid undergoes fermentation. In the presence of oxygen, pyruvic acid is metabolized by cellular respiration, which produces a great deal of energy. Consider the metabolism in your muscle cells. At rest or during light exercise, when oxygen is plentiful, pyruvic acid enters the Krebs cycle and continues to be metabolized through cellular respiration. During heavy exercise, your lungs and circulatory system can't transport oxygen to your muscles rapidly enough, and muscle cells run low on oxygen. The lack of oxygen forces a shift in energy metabolism toward the anaerobic pathway, fermentation. In muscle cells, fermentation results in lactate production. In some organisms, fermentation produces ethanol and carbon dioxide. However, there is no ATP production beyond the two ATP molecules generated by glycolysis. In the presence of oxygen, pyruvic acid is metabolized by cellular respiration, which produces a great deal of energy. Pyruvic acid is transported into the mitochondrial matrix, where it is decarboxylated, that is, it loses a carbon dioxide molecule to become a 2-carbon fragment. This reaction produces two NADH electron carriers. The 2-carbon molecule then enters the next phase of cellular respiration called the Krebs cycle. In the Krebs cycle, the remaining carbon atoms from glucose are incorporated into molecules of carbon dioxide. This process produces two molecules of ATP and a series of electron carriers. The electron carriers essentially transport energy from glucose to the next phase of cellular respiration. The electron carriers donate their electrons to a series of complexes within the inner mitochondrial membrane. These complexes, together called the electron transport chain, use the donated energy from the electron carriers to pump protons into the intermembrane space, forming a concentration gradient of protons across the membrane. The protons flow down their concentration gradient through the enzyme ATP synthase, which is a membrane protein. ATP synthase uses the energy released by the proton flow to produce ATP. From this process, ATP synthase produces an additional 34 ATP molecules, but 2 are used in moving the NADH produced in glycolysis into the mitochondria, thus bringing the net ATP yield from 1 glucose molecule to 36. Let's look at the Krebs cycle and electron transport chain in more detail. The Krebs cycle is a series of reactions that remove electrons, donating them to the electron carriers NAD+ and FAD to form NADH and FADH2. These carriers will contribute their electrons to an electron transport chain that will then generate significantly more ATP than is produced by the Krebs cycle. Each 2-carbon molecule that enters the Krebs cycle is converted to carbon dioxide. Thus, every carbon atom from the original glucose is ultimately incorporated into a carbon dioxide molecule. Finally, 1 molecule of ATP is released for each pyruvic acid molecule that enters the Krebs cycle. Because each glucose molecule contributes 2 pyruvic acid molecules, the Krebs cycle generates a total of 2 ATP molecules for each glucose molecule metabolized. The electron transport chain and the enzyme ATP synthase participate in the final phase of cellular respiration, electron transport. The earlier phases provide energetic electrons in the form of the electron carriers NADH and FADH2. Here, you can follow the fate of electrons donated by NADH. The electrons are passed to the first carrier in the chain and then move down the chain, losing energy as they go. The energy is used to pump protons across the membrane into the intermembrane space. This creates a proton gradient that is a source of potential energy. The last electron acceptor is oxygen, which reacts with the electrons and protons to form H2O or water. The bonding of oxygen with electrons and protons is the reason cellular respiration depends on oxygen and works only under aerobic conditions. ATP synthase allows protons to diffuse across the membrane down their concentration gradient. It uses the energy released by the flow to synthesize ATP from ADP and inorganic phosphate. Let's examine the total energy generated by cellular respiration as it's stored in high-energy molecules. The total includes the ATP from glycolysis, the NADH from glycolysis, the NADH from the formation of the 2-carbon molecule, the ATP from the Krebs cycle, and the NADH and FADH2 from the Krebs cycle. From the energy of all the NADH and FADH2 molecules that enter the electron transport chain, approximately 32 ATP molecules are produced. So, cellular respiration yields a total of about 36 ATP molecules, much larger than the yield, 2 ATP molecules, from the partial oxidation of glucose by fermentation.